So hello student, in today's presentation we will be discussing some other structures apart from the viruses such as viroids, virusoids and the prions, and discussing the viruses I guess is more relevant in today's scenario. As you can see, the coronavirus has created a pandemic in today's world, so I guess it is more relevant to talk these structures. And in addition to viruses, there are some other um, structures are also occur, such as viroid, virusoids and their prions, that we'll be discussing one by one in today's presentation. So first let's have idea how these structures are differ from the viruses. Why cannot we, when we talk about the virology, why we cannot actually use these or use the terminology viruses for these structures? So the differences is, whenever we talk about the viroid consists of the genetic RNA only, and they are apparently very small in the size. They are 240 to 375 nucleotide long structures, and you can see they are naked, means they doesn't have any protein or capsid. Whereas when we compare them to the viruses that I will come to in my next slides, they are differ because viruses contain DNA or RNA as the genetic material, whereas whenever I use the terminology viroid means they contain only the genetic RNA and they do not have any protein code. So just the RNA is an infectious agent. When I say satellite RNA or sometimes virusoids, they are also a genetic RNA, but they are incapable of replicating by themselves. Means virusoids, they are also RNA, but they can replicate inside the host. So how viroids are different from the virusoids? Because virusoids need another help of another virus that we call as the helper virus for the replication. That's why they are put in a different category. So they resides in other viruses and they are infectious in nature. And the prions are only the proteins that are capable of causing the diseases. So they does not contain any genetic material in the form of DNA or RNA, and they are conformationally changed protein. So we can say, we can sum up this slide like viroids, virusoids, and prions are unusual. I use the term here unusual infectious agent characterized by having a very small genome, and in the prions possibly no genome at all. They do not contain any genetic or genetic DNA or RNA. So let's talk each one in detail. So we will start with the viroid. They are very small, covalently closed, circular RNA molecules. They are infectious in nature. They do not contain any polymerase, so they need host-encoded polymerase for the replication, and they causes certain plant diseases that we will discuss later. But just to give you an idea, they cause avocado sun blotch viroid, apple scar, akin viroid, and hot stint viroid. And Theodore Diener is the one, when we will discuss the history, but Theodore Diener is the one who used the terminology viroid, which means virus-like. So I will tell you why we are saying virus-like, not exactly we are putting in the virus category. So one of the example of the viral example, well-known example, is the potato tuber spindle disease. And in the lower bottom part, you can see the normal potato here. They are the healthy potato. And the upper panel is showing the potato with this potato tuber spindle disease. And you can see the elongated potatoes they, and you can see the cracks in between them. And this picture is taken from a review article published by the Hammond and Owens in 2006. So all viroids are inhabitant of higher plants and most cause diseases, and they differ from the viruses because they contain only RNA, whereas viruses contain DNA or RNA, and they do have a protein shell, whereas viroids does not have any protein shell. So that is a difference between viroid and viruses. If we talk about the difference between viroids and prions, that they are different from the prions because prions are made up of only protein and they lack nucleic acid, whereas viroid are made up of only RNA. They do not have any protein code. The smallest viroid known, it contain 220 nucleobase is the small cytoplasmic RNA that is associated with the rice yellow mottle subneo virus, published by the Collins in 1998. And in comparison, the genome of the smallest known virus that is able to cause an infection, they are kilobases, 2 kilobases in size. So the smallest virus is 2 kilobases in size, whereas I told you viruses are very, very small compared to the viruses because they are 274 to 400 something in nucleotide in size. And they cause a disease in one species and do not affect the other species. So how these are originated and how do they spread is they have originated in the hypothetical prebiotic RNA world. As I told you in my previous lectures that RNA is the nucleic acid that was appeared first then to the DNA. So RNA came first and then later DNA came. And they can possesses a ribozyme activity and they are GC rich, which would attenuate the low fidelities of the replication activities, and they are circular. They are covalently circular and do not require start and stop function for the replication. And they cause disease within the plant. So they move within a plant and probably in association with the host protein via the phloem vesicular channel and plasmodesmatar cell contact points. And they seem to form a quasi-species population, and they can recombine. They can have the recombination, and they can spread at least in the commercial crops. So that these sort of the viroids, they cause the economic loss, and they seem to be very dependent upon the activities of the man. And viroid of a particular type are widespread in some areas and they're absent in others. So this slide depicts the difference between the viroids and the viruses. As you can see, looking at this picture, this one shows the differences here, and this one shows the common things. So virus and the viroid, what is the common? They both are infectious agents. They are really not a cellular structure, and they both need host cell to reproduce. The difference is viruses contain the protein code, whereas there is no protein code in the viroids. In the viruses, genetic material can be DNA or RNA, whereas viroids contain only RNA, that is the naked. Viruses infect all organisms, include humans and the plants, whereas viroids restricts only to the infect plants. And the source of the slide is mentioned as a reference here. So we look at the discovery of the viruses. In early 1960s, it was first developed or first identified in the potato, causing spindle tuber disease. Sorry. And in 1965, it was with the collaboration with the Diener, who is the first one to show or use the terminology viroid. In 1968, then characterization of the chrysanthemum stunt and the citrus exhaustis as a non-typical viruses. Here they use term non-typical viruses because they were not exhibiting the characteristics of the viruses. So simply they use the term non-typical viruses. In 1971, Diener demonstrates that the agent is a free non-coding RNA, and that's how the term viroid was used or coined. In 1973, the electron micrograph shows viroids hairpin structure. In 1974, confirmation that the viroids are the non-coding structures. And 1976, electron micrograph shows that viroids are actually closed circular RNA, covalently closed circular RNA. And in 1978, potato spindle tuber virus sequence was sequenced. So if we look at the nomenclature, there are two families that we will discuss later, but the two families, major families are the popsiviroid and avaciniviroid, and these two families are basically categorized based on the replication, as one has the symmetrical rolling circle model, whereas another one has the asymmetrical rolling circle model of the replication. So the Popsiviroidae contain three families: Popsivironi, Episac and Collinivironi, and the genus is as follows, and these are the species. So the some um, examples of the viroids causing the diseases is from the genus Popsiviroids, the potato spindle virus, genus Epsichiviroid, the apple scar skin, the genus Scoliaviroids, Scolius bullemani 1, genus Hostoviroid cause the hop stunt, and the Coquidoviroids causes disease in the coconut kadang kadang, and the genus Epsoniviroids cause the disease avocado sunblotch in the avocado. Here I am summarizing the viroid causing the disease, and this is the host plant. So potato spindle tuber viroid in the potatoes, chrysanthemum stunt viroid in the chrysanthemum is the host, citrus exocortis viroid in the citrus, coconut kadang kadang viroid in the coconut plant, and then avocado sunblotch viroid in the avocado. So just for your knowledge, many viruses can also causes the similar symptoms such as stunting. In the plant stunting, plant get short in the height, and the leaf apinesty means leaf get the leaf orientation is downward, and distortion of the leaf takes place. Fruit distortion takes place, color break, and stem and leaf necrosis, and sometimes even the death of the whole plant. So that is also dependent on the environmental conditions. So let's go with the molecular and biological feature of the viroids. So I'm putting here an example of the potato spindle tuber viroid. And I told you earlier, these are the circular covalently RNA structures. So here it is a small single-stranded covalently closed circular RNA. They do not contain any protein code or the capsid. And there are currently 20 known viroid species that are grouped into two families that I showed you earlier. Pospiviroidy, PSTVD member, and then Epson viroidy, ASVVD member, based on the sequence and the structural homology. In general, member of the Epsoniviroid, they have a narrower host range, whereas member of the Pospiviroid, and for a comparison, description of the host runs of the viroid I will show you in a next slide. But here it is the sequence of the viroid you can see. I will show you later, but it contains 5 domains. Left terminal pathogenicity domain, that is a centrally conserved domain in all the viroid, and then the variable domain and the right terminal domain. Coming up with the, here are the genus you can see of the viroid and the species they are causing the disease and what is the host. So I told you there are two family, Pospiviroid, and the another family is Absenviroid, and you can see the family Popsiviroid contain 1, 2, 3, 4 genus Pospiviroidy, Hostiviroid, Abscaviroidy, and Coliviroid, and they are here causing, like for an example, Pospiviroid causing potato spindle tuber, chrysanthemum stunt, citrus exocortis, columena latent, and the host is potato, chrysanthemum, citrus, and columena. So just let you know, the disease caused by these viroid causes an economical loss to these crops and the plants. And then hostoviroid cause the hop stunt in citrus, grapevine, and prunus species. Apsa, um, Abscaviroid causes the apple skin disease in apple and in pear. Coliviroid causes Coleus blumeni 1 disease in Coleus and Mentha species. Now coming up to the family Absenviroidae, the family contains 3 genus: Abseniviroid, Palameviroid, and Elaviroid. So the Absenviroid causes avocado sunblotch disease in avocado. Palamoviroid causes chrysanthemum chlorotic mottle disease in chrysanthemum, and elaviroid contain eggplant latent disease in the eggplant. So now what factors are responsible for the viroid pathogenesis? So there are 3 factors. Viroid RNA structural element because it contains only RNA. Then RNA silencing. I will show you that the viroid, they have the ability to cause the RNA silencing, and then in addition, host factors are also responsible for the pathogenesis. So coming up to the host factors, cellular proteins, plant phloem proteins, phloem lactin PP2, viroid binding protein where P1 and specific sequence or structural motifs of the host factors are responsible for the viroid movement. Now, as I discussed in the previous slide here, the RNA silencing. So, RNA silencing is something is related to the viroids. So, viroid replicate via double-stranded intermediate RNA. And I'm going to show you in coming up slide, because these are the single-stranded RNA. So, the intermediate phase is the double-stranded RNA, and they're targeted. This double-stranded RNA is targeted by a dicer enzyme, and then it is cleaved into siRNA that are then loaded onto RNA-induced silencing complex. The viroid siRNA actually contains sequences capable of complementary base pairing with the plant own messenger RNA, and then causes the induction of degradation or inhibition of translation that causes the classic viroid symptoms. Okay. In addition to RNA silencing, some viroids are the ribozymes. What does it mean? That RNA having enzyme property, ribozyme. So their RNA have enzyme property that allows self-cleavage and ligation of unit-size genome from larger replication intermediate. So it has been proposed that viroids are escape introns. Okay. So not all viroids are known to be pathogenic, but some are really causes like serious diseases in the plants, and they can be transmitted by the seed or pollen, but they, in addition to that, they may be transmitted by farm implements. An infected plant can show distorted growth, as I show you in my previous slides, and sometimes they can be killed by the viroids. So looking at the genome here, you can see the genome of this viroid. As I told you, it is a RNA, and you can see the single-stranded RNA, but here you can see the base pairing. So single-stranded RNA circle has extensive intra-strand base pairing. You can see here, and the interspurt region you can see as a unpaired loops here, and this one is the base pairing. And in this slide you can see around 80 to 90% of the RNA is base paired to form a rod-like filament, but there are some unpaired regions protruding as a loop. So viroid structures, there are two main groups of the viroids, self-cleaving and non-self-cleaving. Non-self-cleaving viroids replicate in nucleus and fold into dog bone or rod-like structures. All are covalently closed circular RNA, and they are tightly base pair structures, and there are 5 domains coming up with the structures. I will show you the 5 domains. So this is the RNA structures, and you can see the base pairings here, and the 4 domains is coming up with the here. So this is the left hand terminal, so TL showing here. Then terminal right is this one, right hand terminal domain, this one. Then this one is the pathogenicity domain, and then coming up is the central domain, that is conserved among viroids, and then the 5th domain is the variable domain coming up to these structures. So most changes in the viroid here in the pathogenicity domain as well as in the TL domain. So P domain and TL domain, they are responsible to causes the variation in the viroids. Schematic model of viroid structure. Here I have shown you 3 structures. A, you can see the rod-like structure, that is for the potato spindle tuber virus. And then this one, quasi rod-like secondary structures, that is for the avocado sunblotch, the member of the family Epson viroid. And the third one is a complex branched formation proposed for the peach latent mosaic virus. You can see mosaic viroid, and you can see it makes a complex structure compared to these 2. So the now question arises how these viroid replicate. So they contain RNA and it does not code for any protein, and some of the viroid even lack the AUG initiation codon. As you all are aware that the initiation codon is mandatory for the replication. Um, in addition, they might be located in the cytoplasm or in the chloroplast, okay, because these are the site of the replication and they replicate autonomously in the host cell. So the mechanism of the replication involves interaction with the RNA polymerase 2. An RNA polymerase transcribed into messenger RNA by the rolling circle model. Viroid replicates autonomously, and there are three enzyme activities are required for the replication. They require RNA polymerase, they require RNase and RNA ligase. So let's discuss one by one. Here you can see the RNA, and then replication of the RNA takes place by the rolling circle model, and then copying of the, here you can see the second strand, and the second strand displacement takes place, host nucleases processing takes place, and then RNA is, here you can see, being it's exited through this mechanism. So they do replicate by the rolling circle, and there are two types of the rolling circle, asymmetric and symmetric rolling circle. So when I show you earlier, there are the two families in the viroid, and the reason of characterizing these two families is based on the mode of the rolling circle replication. So you can see the member of the Popsiviridae family, they do replicate by the asymmetric rolling circle. So what does it mean, asymmetric rolling circle? Before going into detail, let me remind you, they need RNA polymerase, they need RNase, and they need RNA ligase for the replication. So bear with me here. You can see the red circle here and then blue one. Blue one is showing you here newly synthesizing RNA strand. Okay. So this red circle here is positive infecting circular RNA strand, okay, of the viroid. And this positive circular RNA strand will serve as a template, okay, and then transcription takes place from this template, and it makes this blue one negative RNA strand, okay, and this happens with the help of the RNA polymerase 2 enzyme. Now this transcribed linear negative strand or RNA, negative strand of RNA will serve as the replication intermediate for the synthesis of linear positive strand. Okay, so it will make the positive strand. Now in this stage, host RNA's activity, it needed, because I am saying concatamer. Concatamer means multiple subunit together. Okay, so host RNA's activity cleaves this positive strand into unit viroid length here, and then this unit viroid length is generated by the host RNA's activity, and then with the help of the host ligase activity, these unit length are circularized and then form the infectious viroid, and then this one again serve as a template for the replication. So this is the asymmetric rolling circle replication. Coming with the symmetric rolling circle replication, the difference is in the symmetric one, there are two rolling circle mechanisms are happening, and it occurs in the member of apes and viroidae. So they replicate here, the positive strand of RNA is serving as a template for the transcription of a linear negative strand of RNA. Okay, so in the asymmetric one, this negative strand of RNA serve the template, okay, for the replication, and then RNA's activity takes place, cut it into unit length, and then ligase activity circularize it. But what is the difference here? Instead of serving as a template, this negative strand of RNA, instead of serving as the template, it serves as a direct template for the synthesis of the positive strand of RNA. Okay, and is self-cleaved by the associated ribozyme activity here, and then RNA circularized to form a negative strand circle, and then again our second rolling circle event makes a long linear positive strand in the red one, which is again cleaved by the ribozyme activity, and then it is circularized. So to sum up here, the positive strand of the RNA is acting as a template, okay, and then transcribed the negative strand. Negative strand in the asymmetric model serve as a template, and then RNase activity and ligase activity takes place. Here, what is happening, this negative strand here is circularizing. It's not serving as a template in this condition. It's circularized, and then again rolling circle model happens. Second round of this rolling circle model replication happens, and then this red one, positive RNA strand is being synthesized. Ribozyme activity takes place. It cuts into unit length, and then host ligase activity circularize this unit length. Okay, so that's how symmetric rolling circle replication takes place. So depending upon certain distinguished feature, these viroids have been classified into two groups, group A viroids and group B viroids. Example, avocado sunblotch viroid, peach latent mosaic viroid, and group B again classified into B1 and B2. B1 contains potato spindle tuber viroid, coconut kedeng viroid, tomato plant magoviroid, and B2 contains citrus bent leaf viroid and pear blister canker viroid. So what is the difference between group A and group B viroids? Please bear with me with this table. It may looks very complicated, and this table has been taken from the reference below here. So group A viroid, this group is lacking in centrally conserved region called CCR. If you want me to go with the structure here, you can see when I showed you the 5 domains, okay? So they do not contain the centrally conserved region, okay? So group A viroid lacks centrally conserved region, and whereas group V contain central consensus domain CCR. Group A possesses ribozyme activity, they replicate in chloroplast. Group V, they lack ribozyme activity, they replicate in nucleus and nucleolus. Group A, for viroid replication, they require RNA polymerase, RNase and RNA ligase. वो चीख क्यों रहे हो? और क्या, मैं प्रेजेंटेशन बंद कर उसको, गेट को. Whereas in group V viroid, they require RNA polymerase and an RNase and RNA ligase. Okay. Replication of the viroid require RNA sorry, I think this one is repeating. Here concatenameric multimeric negative strands synthesized. Here they replicate by a asymmetric mode. Okay.